if you're a computer scientist, you're probably going to need some biology knowledge. That's actually very important. And the reason why I say that biologists or biology is so important is because you're building computational models based upon some biological happening, something that happens in biology. And so if you don't understand exactly what that model is, then it's going to be very difficult to build a model where you're actually writing code. And this is actually very important because in the early days of bioinformatics, like back in the 60s and 70s and 80s, and what we know today to be bioinformatics, many computer scientists were building models trying to predict certain types of eventualities in biological systems where they just thought, well, this biology doesn't work this way. That's incredibly inefficient. I'll just do it my way, and that should work. That should be better. And that's because, after all, here we have DNA trying to get to protein. All these other steps seem to be kind of inefficient, so therefore I'll just write this as efficiently as I possibly can, assuming that that's how the biology works. But biology, though, the mechanisms in biology are not. They are not efficient. You know, they're not efficient in the sense that we have, like, you know, computers where you push a button and then something happens. Whereas in biology, you push a button and then this happens, and then something else happens, and something else happens, and something else happens, and then something else happens, and eventually you get whatever you want to happen at the end. And so it's not exactly an efficient system here. And so you do need to understand how those systems work. And once you understand those biological systems and mechanisms, then you can proceed to actually create the models that are working, or that you can work with to actually produce some kind of understanding of the outcome of the model. So you also need, computer scientists also need, for instance, model building skills. Now you're probably thinking that if a computer scientist, I mean, computer scientists should know that, right? They should know model building. Well, not necessarily. There are actually classes or books that you can read that just talk about, building models. Like, is it regression that I should be using over here? Should I be using machine learning? Should I be using some kind of artificial intelligence? Should I be using some supervised, or is it a non-supervised type of model to create the study that I want? It does make a difference. When I first saw this here, my computer science teacher, who is computer science and biology, but bioinformatics teacher, I guess, but they were more of a computer scientist. They were saying that the models that you write for certain biological types of studies are kind of like the tires that you put in your car. If it's winter, you might want to have like winter tires, and if it's summer, you don't need winter tires. You know, you don't need change on your tires. And so it does make a difference into the whole efficiency of how you can drive your car or how you can drive your data. Just quickly, some ideas on careers. There's a professional link that I put up here, so you can kind of like get an idea or a snapshot of like what kinds of careers actually are out there if you're interested in going into bioinformatics. But these are the kinds of career ideas that exist like now, but they always change. There will be more stuff later. I mean, it seems like every day you see there's some new area. But data scientist is probably one of the big things here. A data scientist is someone who looks at data and tries to figure out what that model would be. Like, how do I get some study out of this thing here? And so I think that a lot of these areas are using data science in some way. But research scientists in biotechnology, that's someone who's looking at the data and they're looking at the biology and they're trying to find out whether can they make a model that would understand the biology using this data. It's like they have to put things together. Computational immunologists is somebody who's looking at, again, models to try and figure out how the immune system would work. That's actually a very important area because if you think about how polio was stamped out, polio was a very bad disease that many children had back in the 60s or 50s and 60s, I guess. It's been stamped out of India now. It's no longer there anymore because of certain computational models, which in fact I think that Bill Gates, Microsoft star, one of his research groups and some of his philanthropy was used to create models to try and figure out how you can actually stop, or how do you stop the spread of polio. And so that was a computational immunologist type of area where they were working with this. Anyway, what I'm saying though is that there are many, many different facets and careers out there. And so I don't want you to think that this is just a class and it means nothing when I leave this room. But actually out there though, if you're interested in having a meaningful, challenging career where you can make six figures, which is basically just out of college, maybe you should consider bioinformatics. I mean, maybe now you should consider bioinformatics and save the world from the next pandemic. I mean, no pressure, but I'm sure that you have it in you to find that. You just have to stick with it, and you'll find that thing. Anyway, so let's move on here. This is just some job listings. I just wanted to show you about six figures. This is a bioinformatics scientist working in California. You can see from that the pay rate or the pay scale here, it's 99,000 to 160,000. And you stay in that job for a while, and soon you start making some real money. Anyway, but still, what I'm saying is that the opportunities are there. They're all there. And here's some information about wages. You can see that the wages, if you're not maybe on a salary necessarily, but you're still working as in a lab in some way here, the wages are still getting, they're still getting bigger. They're still getting more and more. The compensation is still getting better. Just some trends you can look at here. How about salaries? You can see that salaries are increasing, but they're not just increasing with inflation, but they're actually increasing because of demand. So it's all there. Anyway, so that's it. I don't want to spend forever talking about these things. You have the slides, and if you're interested, you'll be looking at that, and you can look through some of the links that I put out there to get an idea about what else is available to you in terms of careers. Now, we need to spend some time talking about biology. Now, I know that some of us here might think, oh, biology is just all about memorizing things and understanding strange systems, but actually, though, it's not as difficult as that. If you look at these biological systems, you'll see that they actually do make sense in a way. They actually do make sense. And so I wanted to spend some time just talking about some of these genetic systems and talk about DNA today before we get started here. But one of the things that I find most remarkable, and every time I see this, it impacts me in the same way, and that is that all organisms have genetic systems. They all have genetic systems, which means that you're able to analyze the DNA of seemingly any kind of organism, which is just incredible. I mean, imagine that, though, isn't it? I mean, like you're studying bacteria with a certain type of method, and you can use that same method to study humans. You're studying the DNA in both of these, but that study can still be done with both of those organisms, and that is because all of us, we all use DNA. So it doesn't matter how important we think we are. We're actually very similar to each other. I mean, very similar as people, but also very, very similar as people genetically. There's almost like, there are very, very small differences between us genetically. But like, for instance, between us and bacteria, again, there are great similarities, which are just extraordinary. And when people first saw those similarities, they thought there must be something wrong with the analysis here because I am not a bacteria. But actually, we are very similar to bacteria. One fun fact is that 90% of our cells actually don't belong to us. They actually belong to bacteria, like the flora in our intestines and everywhere else. It's like you have so many different cells inside you which have taken up residency that live inside you. I mean, you're 10% you and 90% bacteria, and that's a good thing. It's called symbiosis, where you're all working together. But what I'm saying though is that in all that mixing and moving around of all that foreign DNA inside you, it makes sense that some of that DNA became your DNA and some of your DNA became its DNA. And so you're kind of now all very similar. And evolution has kind of shown us that we're all very similar by genetics. But one thing I wanted to talk about is that when you see textbooks that talk about DNA, and especially if you don't really know much about what DNA is, the textbook will always show you a picture that looks like this. It's like this picture on the left-hand side where you have this test tube, and if you hold it up to the light, you'll see this nice, twisted ladder or this helix shape inside the test tube. That's not what DNA looks like. That's what the model looks like. But DNA, as it comes in the wild, looks like cotton. It looks like cotton that's like kind of just inside the liquid. Yes, sir. Sorry? Yes, we're surprised that we can even see the DNA. Well, actually, Anthony, I'm glad you said that. I'm glad you said that because my next slide is actually how do you see DNA. You can watch this video. This is actually quite interesting. I've watched this 4 times already. But here is you can actually make DNA visible by moving DNA out of strawberries into solution. And so what you do here is this is a simple experiment you can do at home. You need salt, detergent, and of course some fruit that will have the DNA. In this case, they chose strawberries. But this is from the University of Lancaster, I guess, or Lancaster. And so what they've done is this woman has taken a bag. She's put some strawberries in there, and she kind of squeezed the strawberries around and kind of made them all into mush. Then she added some water, just a cup of water. You can see on her table there's a cup. She added that cup of water. She added just a pinch of salt, which makes which starts agitating, I guess, or bringing the DNA out of the cell where it is. And then she added some very high proof alcohol to this solution with some detergent. And together, the detergent breaks down the cell membrane, and then the alcohol actually pulls out the DNA to the surface. And then when you hold up the cup that she has right now that she's looking at, that cup, when you hold it up after maybe 10 minutes of letting it just sit, you see this white fluffy stuff appears inside there, and that's DNA, and that's the stuff that Watson and Crick were working with. And so it's really not a difficult science to extract DNA. And so what I'm saying is that you can do this kind of work seemingly anywhere. Bioinformatics doesn't have to have expensive equipment necessarily. I mean, you can do it with your laptop. You can do this with, you can extract DNA with a very basic experiment like this, but it's not like a nuclear reactor where you have to have all this expensive equipment to run experiments. Anyway, I do ask you though. I mean, I not ask you, but I do encourage you though to check out this video, because I think that it's actually quite interesting, and you can run this experiment at home. And if you have like any youngsters around you who are interested in like maybe a Saturday science experiment, do it. Do the experiment. Show them. Open their eyes, and you'll see that maybe you'll impact them. Maybe they will become the world's foremost expert in DNA research. Anyway. This brings me to what's called the central dogma of molecular biology. These are big words, and what this means, though, is that a dogma is kind of your central theme. It's the central underlining theme of something. Everything is about this one concept. You might say that the dogma is kind of like every single argument, everything that's written, every single piece of knowledge that's ever amounted in some discipline is there to support some notion, some idea. And in this case, that's what we call the dogma. And the idea in biology is that DNA is then changed into RNA, or the DNA gives us RNA, which then gives us protein. There's three things: DNA, RNA, and protein. That's what's meant by the central dogma of molecular biology. And so every single study that you find in biology, every single thing, none of those studies is going to say anything about DNA doesn't give us RNA, which gives us no protein. They're never going to say that, because that goes against the dogma. The dogma says that DNA gives us RNA, which gives us protein. You're never going to find any paper that's the contrary. So that's kind of like your if you think of this in terms of like an idea cloud, every single publication in biology, every single piece of knowledge, everything that's done supports the notion that DNA gives us RNA that gives us protein, or some part of this thing here. RNA gives us protein, or that protein comes from RNA, or that RNA comes from DNA. It's something like that. It's always in that system. And so what does that mean here? Well, we have DNA, which is on the right-hand side here, that twisted helix. That is the DNA, which then undergoes something called transcription, which then gives us, or you're transcribing something, you're writing something, like the old scribes in monasteries who would write ancient texts down. Those scribes would then take the knowledge from one book and transfer it into another by writing, by scribing. So the transcription is really taking the DNA and kind of changing that into the language of RNA, which is a slightly different language, but still it's a language which comes from DNA. So you're taking the genetic code and then translating that into RNA. And we'll discuss why we do all this stuff in a bit here. And then you have to translate that. That means you're reading your RNA, then you have to turn that into protein. And so that's where the translation comes from. And so this is the central dogma of biology. All of this stuff here, every paper, everything about biology has something to do with this. So, excuse me, that takes me into the idea of like, well, what is DNA then? Here we are talking about DNA. DNA is like they call it the blueprints. It's the genetic code that keeps all of your genes. Inside your cell, for instance, you'll find the nucleus, and inside the nucleus you find the DNA. The DNA is the cell's it's the most precious of possessions. Never mind the cell wall that keeps all the invaders out. Never mind that at all. Never mind the Golgi apparatus. Never mind the mitochondria. Never mind any of that stuff. It's the DNA. The DNA is the number one most precious, most protected thing that a cell has. There's nothing that the cell knows more to protect than the DNA. And so for that reason, you have this nucleus inside this eukaryote cell, which holds that DNA. And so therefore, you have like this cell wall, and inside the cell wall, you have another cell wall, which is where the DNA is kept, and that is like the vault. It's the most. It's there's like the most. It's the most secure area in the cell. And getting back to this idea here, DNA and RNA. Well, the DNA can never leave this cell. It's always this blue stuff inside this nucleus. It never leaves. It can never leave, or it never leaves the nucleus. And so that's where it's safe. And so in order for a cell to copy itself, it has to make a copy of that DNA, and then that copy, the safe thing, it can lose the copy, and that's fine. But that copy is then sent out into the rest of the cell as RNA, where protein is made. But the DNA itself can never leave that nucleus, because if it does, it could be destroyed. And if the cell's DNA is destroyed, the cell cannot live. I mean, it just cannot survive. It loses its instructions for how to make more proteins if there's any damage. If something happens where it needs to make more mitochondria, if something happens where it needs to make more Golgi apparatus stuff, if it needs to make more or repair damages or do some other protein, if it's lost its DNA, it can't do that. It's like, for instance, losing your mind. It's like you can't do anything if you can't remember what you're supposed to do. If something happened where you had a concussion or whatever it is and you're confused, you'll never know how to drive your car. You'll never know how to drive to the hospital and get help. And so it's absolutely essential that you always protect your brain inside your skull, and you always protect your DNA inside the nucleus, because otherwise, without those things, you're never going to be able to find help. Anyway, DNA is actually a very interesting story about how it was discovered. And by the way, if you have any questions, please stop me and just ask. I'd be glad to stop and take some questions. But DNA was discovered by these two here, Watson and Crick, in 1953. But the research that actually gave them the actual what do you call it? The structure. They didn't discover DNA, but they discovered the structure. The research that actually gave them the structure came from someone else called Rosalind Franklin, who you may have seen this before, these X-ray crystallography diagrams. What we're looking at here is when you take a molecule of DNA and then you shine through like radioactive electrons through this atom. When the atoms hit a molecule, when the atoms hit when the electrons hit an atom, they'll bounce off it. So, for instance, you have a shadow, if you will. This is a shadow picture here, according to how radiation, I think it is, or X-rays hit this molecule of DNA. And so what we see now is this X shape in the middle. You can see this kind of like X that's coming together. This is actually all the molecules which have been hitting the molecule, I keep saying molecules, but all the X-rays which have been hitting this molecule head on. When they encountered this helix shape, those X-rays bounced off, and they created a shadow. And where there was nothing, they continued, and so that's where the exposure came from. But Rosalind Franklin's work, though, was actually the pictures that Watson and Crick used to determine what that shape was. Now, it should be said, though, that Rosalind Franklin never got her Nobel Prize, as the other two did. She should have done. And this is one of the big travesties in research, where some of the credit was not given to people who should have received credit. You can read all about this, actually, or you can learn more about this. In fact, I have a video at the end of these slides where it's more of the history of how the discovery of DNA actually happened. And I think that when you see this, you might be, you'll be impressed at all the work that went into this thing without any kind of computers to actually build these models. Well, they had computers, but not like these computers that we have today. But you'll be surprised, though, that her research, though, was the stuff that gave Watson and Crick the idea about what the structure looked like. Otherwise, they had no idea. And she wasn't given credit. And I think that's a travesty in history. And I think that, well, anyway, there's a lot written about that. Anyway, so the DNA is your deoxyribonucleic acid here, DNA. That's basically built out of these blocks or these bases called A, T, Gs, and Cs. Adenine, thymine, guanine, and cytosine. You can see them at the bottom here. Adenine, cytosine, guanine, and thymine. And these are, if you think about it, actually, they're like alphabet characters. And these alphabet characters are the things that are used to spell out the genes. And they live, if you will, in biology in this DNA sequence, and that's how the sequence is. Or that's how they're recorded, if you will. They're recorded in biology by it being inside this helix. There's more to the helix than just this, but the helix, though, actually provides a number of different. The reason why the helix is because the helix actually provides a number of different mechanisms which can be used to copy that genetic code. So, for instance, when one cell is dividing, you have to copy the genetic code in that cell so that the two cells that come about also have the same genetic code. And one thing that we'll be looking at, whether it's today or whether it's on Wednesday, is how this copying actually happens with DNA. And so this structure here actually allows us to have that copying done. So that's why the helix. I mean, back in the early days when people were thinking about what DNA looks like, they were thinking, well, what is it? Is it like some kind of a biological bookshelf where you just store your genes, or is there something else? Why do we have a special molecule that to hold our DNA? Well, it turns out that special molecule is how we copy the DNA. That's like an efficient way to copy. So more on that later. This is just kind of like some ideas about the anatomy, if you will, about the DNA molecule, where you have grooves and minor grooves. The major grooves are like this longer distance between the ravelings, if you will, or the turnings. The minor grooves are, they use these things to describe certain types of, if there's some kind of genetic damage, or they're trying to locate if researchers are trying to locate a particular base, they'll say this is a base which you'll find along the such and such major groove, or however it is. But this is just a vocabulary that they use to describe where these things are. Anyway, that's kind of interesting. But the actual structure itself is actually magical. The DNA structure is kind of like if you have a ladder, like a regular ladder, and then you twist this ladder, just twist it up together. And your ladder has sides that go up and down on each side of the ladder that provide support. But where you put your feet, those are called the rungs. So when you're going up a ladder, you're putting your feet on the rungs. So if you imagine a ladder type of conformation here, then it makes working with DNA much more simple here, where you can have your DNA structure has the bases, or the A's and the T's and G's and C's, actually where the rungs are. They are the places where you put your feet if you're going up if you're actually climbing this DNA ladder. On the sides of this ladder here, the pieces that actually provide the support for the ladder, you have sugars and phosphates. Those are just different kinds of molecules which are actually necessary to make sure that this molecule doesn't pull apart. Imagine, for instance, getting back to a ladder, how necessary it is to have good, strong supports on either side. Otherwise, when you're going up the ladder, the whole ladder could buckle if it doesn't have the supports. And so the DNA has deoxyribose sugars and phosphates along these tracks on the side here that hold the nitrogen bases and keep the structure of the DNA. Now, one thing I will say, though, is when we go through this some of these slides here, all these chemicals, all these attractions, all these atoms that are involved in basically holding together a piece of DNA, why do we need so much? Why do we need the sugars, the phosphates? Why do we do all that stuff? I mean, isn't it basically a safe environment where the DNA doesn't get pulled around that much? Well, no, actually it isn't. It's extremely turbulent inside cells. I mean, just nodding your head like back and forth like this, you're actually moving your cells around, and inside your cells you have DNA, and that DNA is being kind of swished around in liquid, and of course it's in liquid, so it's not going to get destroyed, but it has to be strong enough to be able to deal with those kinds of things. Now imagine, for instance, imagine snapping your fingers like that. You're going to be kind of, you're really subjecting your skin at a micro level to a lot of stress, and underneath that you have DNA which is even smaller. Imagine how rattled that DNA gets. But actually, though, in spite of all that turbulence and those stresses, this chemistry actually holds the whole thing together. And so your ladder, getting back to this, is actually held together by these sugars and phosphates. So what do I mean by these sugars and phosphates? Well, here we have a molecule. This is like if you take your ladder like this, and I just like cut out two little pieces, well, not two little pieces, like one little between like around the rung here. This base area, what I'm looking at right here, this nucleobase, that is the rung. That's where on the ladder where you put your foot. So this other side over here, this D-ribose sugar and this phosphate group, this represents the sides of the ladder, one side of the ladder. The idea is that each side of the ladder, each side of the helix has the same chemical compound, and then it's connected to a base in some way. But this is the side here. So we're going to talk about this for a second here. The deoxyribose sugar has 5 carbons. Now, if you've had molecular biology or if you've had organic chemistry, then this is like old news to you, about these carbons. But carbons in this case here, in this round purple spaceship-looking object, every time there is a corner that's being bent here, like at the 1 prime, 2 prime, 3 prime, 4 prime, those represent in that purple molecule, in that ring as they call it, those are where the carbons are. Now we don't draw our carbons with like a C inside. We could do, I guess. We don't actually draw them in molecular biology or in organic chemistry with C's, but rather because there is this angle that exists like here, that angle actually is the carbon. So each carbon has a name. We call them first carbon, second carbon, third, fourth, and fifth carbon. Or we call them the first, or the first prime, the second prime, third prime, fourth prime, and 5 prime. You need to know those primes because that's like your address inside the molecule where that carbon is found. And in this model, the nucleobase or the base, the A, the T, the G, or the C, that is supported by the first prime carbon. It's on this first prime carbon here. The phosphate which is this phosphate group over on the yellow thing here, that's going to be supported by your 5 prime. So you're getting this kind of notion here that each carbon in this ring has a specific function to deal with, and it does. And so you have each of these carbons has a name. Let's move into this over here. This is another picture here. Now we see the sugar and the phosphate group on both sides of the helix. It's just a ladder again. But where we see and I do apologize if you don't see the colors very, very well. The orange is the deoxyribose sugar, and the green is the phosphate, which is down here. But the sugar though is that purple thing that we saw. It's this thing over here. That purple thing, that molecule, that ring, fits into this chain looking like this. And you'll see that between the sugars is a phosphate group. So if you think of these in terms of maybe like Lego pieces, I know that Lego pieces sounds like, I still like Lego, but whatever. If you think of these in terms of like Lego pieces, you stack the Lego pieces together in this particular order where you have a phosphate, then a sugar, and a phosphate, then a sugar, then a phosphate, then a sugar. That becomes each rung holder, or each side of your ladder. So yeah, it's actually kind of it's an interesting. I mean, this is actually kind of an interesting science here. I don't want to spend too much time on the organic chemistry, but there is a whole lot of information when you look at these types of diagrams. When you look at some of these molecular structures, it gives you ideas about, for instance, how DNA actually functions as how you can copy DNA, or how you can use DNA, how DNA is transcribed, for instance. Let's see, the sugars in this diagram here, you can see the sugars, the D-ribose sugars, those are the things that actually hold on to the bases. And so if you see in the middle of this diagram on the right-hand side, the A's meet up with the T's, the A's are connecting with T's, and so the A is being held in place by a sugar on one side, and the T is being held by a sugar on the other side. But it's important to notice that the rungs are in different directions. In other words, the molecule is drawn on the left-hand side where the sugar has the point, the steeple, I guess if you will, on the top, whereas on the right-hand side of that illustration, the steeple is on the bottom. I can't quite do this, but it's on the bottom. Otherwise, you have a straight top. And so you can see now that you're building always where you have a protein, a sugar, the phosphate, then sugar, a phosphate, then sugar, then phosphate, then sugar. But it's like you're not just connecting that phosphate to any old carbon, but rather if you look at the left and right-hand side, one is upside down from the other, but it's still being built, that phosphate group is still being built on the same carbon. So that's important because they didn't realize this back in the 50s. They were thinking, oh, well, does it matter which carbon we're using? It does. And these are important facts, because if you're making a model that's trying to work with DNA, to try and work with, to try and, to try and figure out how DNA is transcribed in silica, then you need to know kind of like how the phosphates and the sugars are playing a role in the actual copying process. We'll talk about that in a bit. One thing that's important, though, is when I see these base-to-base bonds, what that's saying is that A's and T's attract each other. A and T. G's and C's attract each other. You never see G attracting to A, and you never see T attracting to G or C. It's always A's and T's and then G's and C's. Now you can remember that, if you will, because when you look at the G and C down here, they're both round, right? They look round. G's and C's as letters are round. So that should give you some kind of, that's your muscle memory right there. That's how you know that G's and C's attract. And when you look at A's and T's, for instance, A's and T's are both kind of, they're letters made out of straight lines. And so that should give you some ideas about how these things actually come together. I say this because, I mean, actually I was at I went to this quiz show thing when I was doing my master's. There was like this quiz show night that we had. We used to go, and everyone on campus would go to this thing. It was like in the student center, and you could basically win a free lunch. And that was really why we went there, because we were all starving students. And one of the questions that they had, and it just happened to be that I was there. One of the questions that they had was, okay, true or false? G's attract T's, G and T. And the person who was next to me was like, yeah, that sounds right. Sure, that sounds like a good thing. And actually, I won this, not because I also from the bioinformatics, but the first thing that came to my mind was, how could G's and T's attract each other? Because G is a round letter, and T is a letter made out of straight lines. It just doesn't work. So I won this. I won a free lunch, which was actually a hot dog and a little tray of chips. It was such a wonderful moment. I wish you guys could have been there. Oh man, just reliving those days. Oh. Okay, we're kind of running short on time here. We have 2 minutes, and so I don't want to take up too much. We're gonna go back to these slides on Wednesday. That's typically how things are going to be done in this class. We'll have slides that come out on Monday. We'll do what we can for Monday, and then on Wednesday we'll complete those slides, and then on Friday we'll probably have a different set of slides. And so we're going to be kind of like, there's always going to be some kind of rollover. But one thing I wanted to say, though, is all of this information here is saying that we say that A's and G's are purines. And by that, we're saying that the chemicals look like two rings together, whereas if you have a pyrimidine, which is a thymine or a cytosine, it's basically one ring. And so this is actually important because that's what the chemical looks like. The chemical looks like, the molecular looks like. That's how and the chemistry will follow from this molecular structure. And so by that, what I'm saying is that a purine and a pyrimidine actually attract each other. That's how the A's and the T's kind of function. So you have to have a pyrimidine, I'm getting myself confused here, a purine and a pyrimidine, which come together. But one thing I'll say to you, though, is that if you look at how the A's and T's connect, there's only two bonds in the picture in the orange above. But when G's and C's connect, there are three bonds. And so what you should be getting from this slide is that 3 bonds is better than 2 bonds. That means that GC bonds with 3 bonds are stronger bonds than 2 bonds, which is made out of the A's and T's. You can try this experiment too. Just take 2 fingers and like put your fingers together and see how much stress you can pull before, if I can get my camera right here, before before it breaks up. But if you have 3 fingers, it's stronger. You can hold things together stronger. You hold your hands together stronger when you have 3 fingers working than 2. And so that's really how you can remember this. So this purines are these 2-ring structures, and pyrimidines are these 1-ring structure. But A's and T's have 2 bonds, and G's and C's have 3 bonds, and that's all because of the way the molecular structures are. Oh gosh, it's already 11:40. Oh, I'm just getting myself fired up here. I'm so excited about this here. I'm just getting so Anyway, I'm going to break it here. I think we'll stop for now. We'll come back to this on Wednesday. But one thing I will say, though, is that we do have lab today, so I will see everyone at lab. I think that's, what time is that? 2:30, is it? 2:15, right. I will be there. It is online. We're not going to be meeting in this classroom. But we're going to be meeting online. There is, I'll just give you a heads up about what we're doing. We are going to be spending some time to install some software, Docker and Git, which we'll be using for this class here. And so please make sure that your computer is well, you have to have your computer, but make sure that you're able to install things on that computer. And that's it. I will see you all at lab. This is going to be a great semester, guys. I'm looking forward to this. This is going to be so exciting. Thank you for coming.